When Darwin first proposed his theory of evolution and his mechanism of natural selection, one of the first things that people's minds leapt to was the question, what does this idea say about us? And this is a very powerful fascination that everybody in the public has with the idea of evolution. If we are, as we seem to be, evolved beings, what does that say about who we are? I'd like to address that question a little bit in this lecture, but not without saying a few important things first. It is true that all features of every organism are the way that they are because of their evolutionary history. And of course, human beings are organisms, so that includes our features. But it is not always straightforward. In fact, it's not always possible to figure out why certain features that human beings have are the way that they are. In particular, the methods of evolutionary biology are most useful when you're looking across a broad range of species, say when you're thinking about a characteristic that has evolved many times. They are not as powerful when you're thinking about something that happened once. So broad trends are easier to address using evolutionary biology than the particulars of a single species. And of course, when you're talking about human evolution, you're talking about one species, and a lot of things that may have happened in the evolutionary history of that one species may not be generally what you see in the rest of all other species. Evolutionary biology, in other words, can tell us what's most likely, but what happens in the evolutionary history of one particular species may not be, or may not have been, that most likely thing. There are a number of tools that, in fact, you can't apply at all to a particular trait, that you have to have a big sample size. You have to have a trait that has evolved multiple times in multiple different species before you can say anything intelligently about how it is likely to have come about. But the most problematic part about trying to apply evolutionary biology to human beings is that it has been done very badly in the past and with some really horrible results. And it suggests that what we often see when we try to apply evolutionary biology to human beings, what we often end up seeing is we see evidence that confirms our own biases. We are very prone to misinterpreting evidence in ways that tell us that what we already thought is right. And there are some terrifying examples of that in the history of evolutionary biology. Perhaps not surprisingly, evolutionary biology was used to prove scientifically that races other than the white race were inferior. It was used to promote eugenics, which is the idea that we should all work to improve the genetic fitness, the genetic goodness, or the genetic righteousness of the human race. And that meant not allowing people who were considered to be undesirable to reproduce. Some really horrific things were done in the name of science, in the name of evolutionary biology. And more than anything, what I want you to get out of this lecture is that you cannot try to apply science to human beings without looking very, very hard at your own biases, the baggage that you are bringing to trying to interpret evidence. Because if you don't do that, and if you don't do it well, what you're going to end up doing is simply seeing what you already wanted to see. One other problem that we have when we try to apply evolutionary biology to human beings is that a lot of the characteristics that we see in human beings aren't genetically based. They're cultural. There are many things, like for example how we dress, that are strongly influenced by cultural. In fact, there are some traits in human beings that are entirely generated by culture. And these, of course, are not traits that are going to evolve in the standard biological evolution kind of way. Nonetheless, there are some things that we do know about human evolution, about how the mechanisms that you've been learning about have led to the species that we are today. And I'd like to talk about some of those. But the whole time I talk about this, I do want you to keep these caveats in mind, is that it is a very delicate business to try to apply evolutionary biology to human beings, partly because we often want to fool ourselves about what the evidence really says, and partly because when you're dealing with a single species, the predictive power of evolutionary biology is much, much poorer than when you're dealing with lots of them. There are, however, some things that we know for certain. What's one of the most interesting things, I think, is up until quite recently, in geologic terms, there were a number of different hominin species on Earth. And hominins are the group that basically led up to modern, anatomically modern human beings. And you can see a whole range of them existed at various different times over the last few million years, and some of them actually existed almost into what we think of as historical time. The discovery quite recently of Homo floresiensis on the island of Flores suggested that we're almost, this other human species existed almost up to the point where human culture, our culture, had developed to the point where we're writing things down. And that's a remarkable thing to think about. One question you might ask yourself is, why is it that Homo sapiens, the species we belong to, is the only one of these various different hominin species left? And the answer is probably that we simply outcompeted all of these other lineages. Human beings are incredible competitors ecologically. It is remarkable the number of species that seem to have been driven extinct by human beings just because we compete for the same food, for example, that they do. And human beings had a lot of advantages over a lot of these other hominins. Anatomically modern human beings had a much more advanced use of tools, including fire, and most importantly, their social interactions are extremely advanced. And these are both things, but especially the social interactions, that we want to be looking into when we look at the evolutionary differences between human beings and other animal species. So what are the differences between human beings and other species of animals that make us really human? Well, some of them are sensory changes. For human beings, vision is very important. And there are other senses that have been lost as a result. So for example, if you take a look at this chart here, what it's showing you is the proportion of olfactory receptor genes that have become pseudogenes. That is, they have ceased to function. The baseline is somewhere around here, somewhere around 16, 17, 18%. That's typical for most mammals. And the lemurs and monkeys, most monkeys here, have about the same fraction of their genes, of their olfactory receptor genes, inactivated. There's one exception here, that's the howler monkey, is particularly interesting. Notice what happens when you get up into some of the monkeys and apes that are more closely related to human beings, though. That percentage goes up much higher. And in human beings, half of the olfactory receptor genes have been inactivated. What's going on here, the reason that this graph looks the way that it does, is this. In this lineage here, leading up to all of these species, three-color vision was acquired. All of these species down here have two-colored vision, with one exception, that's the howler monkey. Basically, in a nutshell, the story this tells us, when we apply this phylogeny to this information here and we reconstruct the ancestral traits of organisms on this phylogeny, what we see is that when more advanced color vision evolves, olfactory receptors are less necessary. And so when mutations occur that inactivate some of those receptors, Natural selection does not act against those mutations. In other words, there's not enough selection pressure to maintain a really strong olfactory sense. And so this is why your main sense for understanding the world around you is visual, and compared to most other mammals, you have a really, really bad sense of smell. Structural changes in the skeleton also have made a big difference in the way that we interact with the world. One of the things that human beings can do that the other apes really cannot is to walk upright on two legs comfortably. Gorillas, chimpanzees, and some of the monkeys, in fact some of the lemurs even, can walk on two legs, but it's not particularly comfortable for them to do so, and they certainly can't move that way at speed. The hip joints of human beings, basically the way the hip articulates with the femur here, is different than the way that it is arranged in the other apes, and that allows us to stand upright all the time and move, if not nearly as quickly as many other animals, move more quickly than the other apes can while standing on 2 feet. It's not clear whether that preceded or followed the evolution of manual dexterity, but certainly being able to stand up on 2 legs frees the hands for doing a lot of other things. And in general, human beings are much more dexterous with respect to their hands and fingers than any of their close relatives. But far and away, the biggest differences between human beings and other primates are in the brain. This graph here shows some of the evolution that's happened over just the last few million years in the lineage that led to anatomically modern human beings. Ardipithecus ramidus, which is, an ancestor somewhere along the ancestral line of modern humans, only 4.5 million years ago, had a brain that is indistinguishable in size from the brain of a modern chimpanzee. And that was true of all of what are called the Australopithecines, shown here in various shades of orange and yellow. However, very quickly, about 2 million years ago, brain size changes rapidly in the groups of species that are most closely related to our ancestors. Now we're getting up into the genus Homo. And brain size goes from something like 300, 400, 500, which is what it was, 300, 400, 500 cubic centimeters, which it was for the Australopithecines, up to 900 for Homo erectus, 1200 for Homo heidelbergensis, and a range that's well above that for anatomically modern human beings. The brain size of the average modern human being is over 3 times as large as the brain size of the average Australopithecine or the average chimpanzee. Now, brain size does not correlate exactly with intelligence, but the massive increase in the size of the brain does seem to be correlated with a lot of the other advances in brain structure that make human beings capable of much more complex thought than other animals are. So here's an approach that's based on what you already know about the molecular clock. It's called the relative rate test. If you've got any two species that are more closely related to one another than they are to some third species, then obviously these two are going to be equally closely related to this one. So if A and B are in a clade, they're going to be equally closely related to any organism that's outside that clade. If you've got genes that are evolving in a reasonably clock-like fashion, then when you compare the DNA sequence of this species B, to the species C, they ought to be different by the same amount than the DNA of this species is from this one. Okay, so for example, let's suppose that these tick marks denote the mutations that happen along these various different lineages. If this gene is ticking reasonably regularly, you'd expect the difference between human and rhesus monkey to be the same as the difference between chimpanzee and rhesus monkey. Okay, you can turn the logic of this around though. All else being equal is something you can test for. If you compare this difference to this difference, and you find that they're not the same number, then what that implies is that the gene has not been behaving like a molecular clock. Or rather, more specifically, what it means is in one of these lineages, leading away from this most recent common ancestor, one of the genes have been evolving faster than in the other. And so, for example, if this were 10 and this were 10, that would tell you that this lineage and this lineage, the gene that you're looking at, hadn't been evolving at the same rate. But it might be that what you'd see instead is that you'd see that there have been a lot more changes along one lineage than the other. And so you might see 14% here and 10% here. In other words, what the relative rate test does is it tells you something about how rapidly these two lineages have been evolving since their most recent common ancestor. This is interesting when it comes to human evolution because you can look at the sequences of genes that are involved in the brain. And that's just what some researchers did. They did the relative rate test for exactly those three species. They looked at the lineage that led to humans versus the lineage that led to chimpanzees by comparing them to an outgroup, which in this case was the rhesus monkey. And they did this for different groups of genes in the genome. So for example, here are some genes that are involved in the blood. And when they compared the human to the rhesus, and then they compared the chimp to the rhesus, they found that the differences they got in those two comparisons were the same. In other words, since the most recent common ancestor between chimpanzees and humans, these two lineages have been evolving at the same rate. When it comes to genes that are involved in the liver, the human genes appear to have accumulated a significant number more differences. Human genes involved in the liver have been evolving at about 130% of the rate that they have in the lineage leading to modern chimpanzees. What's really striking, though, is when these researchers looked at the genes involved in the brain. The genes leading from the most recent common ancestor of humans and chimpanzees up to anatomically modern human beings have been evolving at a rate that's 5 or 6 times faster than those same genes leading from that same common ancestor to modern chimpanzees. That is a tremendous difference, and it suggests that an enormous amount of evolutionary change, probably a lot of it under positive selection, has been happening in the lineage that's led to humans. When you look at the exact genes that are involved in these changes, what you find is it's not the proteins that are involved in the brain so much. The stuff that a chimpanzee brain is made of is exactly the same stuff that you find in a human brain. But the genes that regulate how brains are put together have changed most often in the lineage leading to human beings. In other words, most of that 5 or 6 times increase in the rate at which things have been evolving has been due to regulatory genes. The regulation of the way the brain has been put together has evolved extremely rapidly in humans. This has led to human beings having a brain that is really radically different than the brains of other organisms. One of these specific regulatory changes that researchers have identified is that there were changes that allowed glucose to be delivered much, much faster to the brain compared to the glucose delivered to the brains of other species. And that's tremendously important for our ability to use our brains, because our brain is this really, it's an energy hog. 20% of the calories that you take in are actually used by your brain, despite the fact that it is only about 2% of your body mass. That means that relative to other tissues in your body, your brain is chowing through calories at 10 times the normal rate. So the ability of the body to deliver glucose much, much faster to your brain, that regulatory change was absolutely necessary for you to think the way that modern humans are able to think.